Hello, everybody. Welcome to Thursday, September 30th, last day of September. Couldn't be better. I guess it could be if you were here with us. That would be great to have you here today. But unfortunately, you are at home, absent or otherwise, so I'm here bringing you what you're doing today. Luckily for today, it'll be kind of similar to what they're doing in class because a lot of the stuff they'll be looking at, they're not actually using the microscopes, but rather a microscope website that you'll be able to access at home just as easily as they would here. So without further ado, first off, make sure that you do your bell ringer every day just to get in the habit of doing that. And then we're going to open up our cell investigations day number 2, which is the same as day number 1. So you should hopefully have day number 1 completed, looking at our 2 specimens. Remember, for those specimens, I sent you that absent learner's guide. It is also on Schoology in our folder. Right there, the absent learner's guide cell investigation document. That's where you can find all the information that you'll need to know for the stuff since you've been gone. So there's day 1 and day number 2. And then day number 3, it says use the website linked in the investigation to work on day number 2. That's all you need to do for day 2. So let's take a look at what that is. I've also put it right here in the Schoology folder, cell investigation website day number 2. But it is also right here on the instructional page. Pop this bad boy open, and then you'll get to here. And you'll see that there's some lovely pictures of cells. We have number 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 9 things to look at today when it comes to this stuff. So what you're going to be doing, this is back on the slides, is that you're going to be filling out this checklist, which should look familiar because you had to fill these things out for these specimens from yesterday. You got to check whether it's living or non-living. Remember, that is if it is a cell or not a cell. Cell would be living. Not a cell would be non-living. If you answer it was non-living, you do not fill out the rest of the chart because you can't. Because it's asking about cells and you already said that it doesn't have cells. So you can't really do that if it doesn't have cells to it. If it is living, though, you'll have to go through and do all of our other things, our unicellular, multicellular, prokaryote, eukaryote, square, round, membrane, wall. To it. Remember, for our unicellulars, those would be things that are kind of separated out from each other that doesn't look like they connect really to it. And the multicellular stuff would be if things are clumped together, and there's a lot of them too there. The prokaryote, eukaryote, remember our pro no, you do. So if it has a nucleus to it, it would be a eukaryote, which is that dot in the middle of it. Let me draw. I have pictures up on the board for this stuff, like on the whiteboard that I drew. So let me just show you basically what I drew for them and show you it. Bloop. So there's a little prokaryotes are going to look like this. It's going to still have the kind of membrane or wall around them, but they do not have the big blackened dot in the middle because that big blackened dot would be the nucleus. Eukaryotes, on the other hand, would. See how they got that big dot in the middle. As far as unicellular and multicellular go, unicellular, and then you go like that. Kind of draw some stuff up there. This would be unicellular. This one would be multicellular. Our unicellular example, there'd be some clear separation between them. Whereas our multicellular, they would be almost like bricks in a wall where they're kind of stacked and connected to each other there. So that'd be unicellular versus multicellular going on there. And that's all that I have as reference for them on the boards that you wouldn't be having since you're at home. You can really easily tell, hopefully, if it's square or round, which the square or round goes into having it being a membrane or a wall. If you don't remember which one makes a square and which one makes a circle, that should be in our notes. One of them is more related to the wall, the other one's more related to being circular. to it. But you're filling out the checklist for each one. I will say make sure that you're on the right page as you're doing it. If you look right above my head, you'll see that it says online number 1, number 2, 3, 4, 5, 6, 7, 8, 9. But then the actual slide doesn't change besides that number because I'm asking you the same 5 questions about each one to it there. At the end of it, you have 4 questions to answer. These are all about things from the checklist to make sure that you understand what we're doing with the checklist. What is the difference between a pro and eukaryote? What is the difference between a plant or an animal? How are you able to identify the nucleus? These are just questions to get you to be able to tell me that you clearly understand all the things that you are putting down in the checklist for it there. Now, back to the things that you're going to be looking at. When you get to day 3, you stop. Before I go back. The things that I have on this website, I give you some hints as far as things go. You have 9 things to look at. 3 of these are going to be non-living, which means they are not cells. So you should hopefully have a understanding of what a cell kind of generally looks like. Some of these should not fit in that categorization. 2 of that means the other 6 are going to be cells and are going to be living. Which 2 of these are going to be animal cells, which means they're going to be round and they have a nucleus in the middle of them. 2 of them are going to be plant cells, which means they're more squarish in shape and will have a nucleus inside of them. And then 2 of these are bacteria, which means that they're going to be more roundish in shape because, remember, round is kind of animal / bacteria, whereas square are more plants and fungi. But 2 of these are bacteria, which means that they will be more of that roundish in shape, but they will not have a nucleus. They are blank in the middle. They might have some other stuff going on inside of it, but they are generally just blank in the middle. There's not that clear dot that is in the middle of it. Like this, you can see clear dot. For a bacteria, they would not have that clear thing smack dab in the middle of them, that big old nucleus thing to it. So, if you have any questions, just let me know. Hopefully you're able to keep up with things. Like I said, today's at least a day that was all meant to be online anyway, so it kind of works out well for you since you can't use the microscopes at home. It works out kind of nicely for you. Hopefully, though, you are back with us sooner rather than later so you can actually get to use some of these things. And I hope you're doing well, staying safe there. If you have any questions, email me up. But I've gone through all the things that you'll need to do for the microscopes day number 2. So, with that, I will hopefully see you tomorrow, if not next week. Have a good rest of your day. If you have any questions, email it up. Otherwise, bye.